Hello, I'm Dr. Senkuba Eric. Today, I'll be taking you through one of the most fascinating aspects of human physiology, the special sense of hearing, focusing on the structure and function of the ear as well as the neural mechanisms of auditory perception. Physiology of hearing, the human ear. The human ear is anatomically divided into three parts: external ear, middle ear, and inner ear. These function together to convert sound waves in the air into electrical signals that can be interpreted by the brain. External ear. The external ear consists of the pinna, auricle, and the external auditory canal. It functions primarily to collect sound waves and direct them toward the tympanic membrane, also called the eardrum. Middle ear. The middle ear is an air-filled cavity that contains three ossicles, the malleus, incus, and stapes. These small bones act as a lever system that amplifies sound vibrations. When sound waves strike the tympanic membrane, it vibrates, and this vibration is transmitted sequentially through the malleus, attached to the tympanic membrane, to the incus, and then to the stapes. The stapes footplate sits on the oval window of the cochlea, and it transmits the mechanical vibrations into the inner ear. The middle ear is also connected to the nasopharynx via the eustachian tube, which equalizes air pressure on both sides of the tympanic membrane to ensure efficient vibration. Inner ear, cochlea. The cochlea is a fluid-filled, snail-shaped structure in the inner ear. Within it are three compartments. Scala vestibuli contains perilymph. Scala media, cochlear duct, contains endolymph. Scala tympani contains perilymph. The scala media houses the organ of Corti, which is the receptor organ for hearing. It rests on the basilar membrane, and above it lies the tectorial membrane. When the stapes moves the oval window, it creates a pressure wave in the perilymph of the scala vestibuli. This wave travels through the scala vestibuli, displaces the basilar membrane, and eventually dissipates through the round window at the end of the scala tympani. Frequency discrimination. The basilar membrane is tonotopically organized. The base, near the oval window, is narrow and stiff, responds to high frequencies. The apex is wider and more flexible, responds to low frequencies. Different parts of the membrane resonate with specific frequencies, creating a spatial encoding of sound called tonotopy. Hair cells and mechanotransduction. The organ of Corti contains two types of hair cells. Inner hair cells, primary sensory receptors. Outer hair cells amplify mechanical vibrations by changing their length in response to sound. Each hair cell has stereocilia embedded in or near the tectorial membrane. When the basilar membrane moves, it causes a shearing motion between the hair cells and the tectorial membrane, bending the stereocilia. This mechanical bending opens mechanically gated potassium channels via tip links, allowing K+ to enter from the endolymph, which has an unusually high K+ concentration. This depolarizes the hair cell, leading to the opening of voltage-gated calcium channels and the release of neurotransmitters, likely glutamate, onto the afferent fibers of the cochlear. Nerve. Neural transmission. The auditory signals travel via the cochlear division of the vestibulocochlear nerve, cranial nerve 8, to the cochlear nuclei in the brainstem. From there, the pathway includes superior olivary complex, for binaural processing and sound localization. Lateral lemniscus. Inferior colliculus, midbrain. Medial geniculate body, thalamus. Finally, to the primary auditory cortex and the superior temporal gyrus, Brodmann areas 41 and 42. Protective mechanisms and adaptation. The tensor tympani and stapedius muscles contract reflexively in response to loud sounds, the acoustic reflex, reducing transmission of vibrations to the cochlea and protecting it. The outer hair cells can undergo electromotility, changing their length to fine-tune the sensitivity and frequency selectivity of the cochlea. Summary points from Gayton. Sound intensity is encoded by the rate of firing and number of neurons activated. Frequency discrimination is due to spatial mapping on the basilar membrane. The endocochlear potential, approximately +80 mV, in the scala media facilitates K+ entry into hair cells. Damage to hair cells, especially by loud sounds or ototoxic drugs, is usually permanent due to poor regenerative capacity.